during the break, I was on my tablet just going through a social media site I use, and the first picture I came across was this rather impressive picture of the Milky Way, actually from New Zealand, not Greece, but the idea that the word galaxy really comes from somebody looking up at the sky and seeing this and picturing it as spilled milk or something like that. Sort of where that came from. Anyways, to continue on, the end of the digestive tract, we're looking at this large intestine. And there's not a whole lot to say about the large intestine. So this will be somewhat brief, I hope. I went through the parts that make it up last time. Histologically, there's not a whole lot of terribly interesting stuff to say about it. This drawing is fairly similar to the drawing of the stomach. Just the inside surface is relatively flat with a lot of openings for glands. I didn't talk about the intestinal glands in the small intestine. What's in large intestine is somewhat similar. However, in the large intestine, there's a very high count of goblet cells. They're in the small intestine also, but if you look at the histology of the large intestine, it's almost like every other cell is a goblet cell. Goblet cells make a very watery mucus to secrete that out, and that's about all that the large intestine really does as far as secretion is concerned. The point of a large intestine is really to take the indigestible material of the food that we had, that we've gotten all the nutrients out of, and to get every last little bit of stuff that we can out of it, which is largely helped by the microorganisms that live in the large intestine, breaking down that indigestible material, which is what we often refer to as dietary fiber. It's carbohydrates that we don't have the enzymes to break down, namely cellulose. So those organisms can break it down. We get no caloric benefit from that. It's strictly their food source. But in doing that, sometimes they can release certain micronutrients, meaning vitamins and minerals, that we might need. So we can get a little bit out of it there too. The large intestine, is a series of kind of bag-like regions called haustra. Haustrum is singular. It's sort of like a little muscular pocket that the food stuff fits into. The muscles will contract and kind of squeeze it to get whatever last little bit of water that might be inside of it out. Then peristaltic type contractions will come along and move the food through this large intestine. The food doesn't just move from one haustrum to the next. It'll move from one haustrum down a ways with each peristaltic wave. I don't remember exactly where I read this, and since I can't remember where I got it from, I can't verify it, but I think I read somewhere that the peristaltic waves in the small intestine come at about half-hour intervals. Again, I can't entirely verify that. I don't remember where it came from, but I haven't read anything to suggest it's not true. And so the food stuff in the large intestine will be pushed along at about that rate. As it's passing through, the microorganisms can do their job to digest what they can. We can get the last little stuff out of it. Eventually, there's not a picture of it. I thought there was a separate picture showing the rectum and the anus, but apparently there's not. Eventually, the food stuff moves to that point, and it's then going to be eliminated. Elimination of solid waste from the large intestine is referred to as defecation. It's a reflexive action. When stretch receptors in the rectum, which is essentially where the colon comes to an end, the sigmoid colon comes to an end here, that's the rectum. As the wall of the rectum starts to stretch out, those stretch receptors send a signal into the sacrospinal cord, and the signal coming back through the parasympathetic fibers will cause contraction of the muscularis of the rectum, pushing the feces out. There's a smooth muscle sphincter at the opening of the anus called the internal anal sphincter, which relaxes when the parasympathetic system pushes material out. That sphincter can be contracted by sympathetic input, such as during the fight or flight response, but probably for the most part isn't terribly contracted, because while there's an internal anal sphincter, like I just said, there's also an external anal sphincter, which we have voluntary control of. Having voluntary control over a sphincter means that that sphincter is made out of skeletal muscle. And if it's not a socially appropriate time for you to void your bowels, I suppose, then you have the control over that external sphincter so that nothing is going to leave unexpectedly. There must be a point, and for the digestive system, I've not really read that this is the case, but it must be that at some point the large intestine can get can overcome that voluntary control. You don't want the rectum to fill with feces to the point it would burst. So there must be some sort of ability of the autonomic system to overcome the voluntary control of that sphincter. I'm not positive of how true that might be, just because it's not something that's mentioned a whole lot. The same thing, however, occurs in the urinary system, which we'll be talking about later. And hopefully I'll get to this part of the urinary system before we leave today. And your urinary system can overcome the voluntary control of the external urethral sphincter. I think something like that must be present in the digestive system, but it's probably not quite as strong of a system. That's largely what the large intestine's about. Collecting the undigestible material, getting the last little bit of stuff out of it, and then eliminating it through the defecation reflex. The next section, I thought a little bit about going into the next section before talking about the large intestine, but I decided to just go ahead and follow the order of the book here. The next section is looking at the accessory organs of the liver and the pancreas, and then the gallbladder. Really, when I talk about these, I like to say liver/gallbladder, because those two are connected and very related to what they do, and then the pancreas is separate from that. This just shows the locations of those three organs. The liver is making up most of the upper right abdominal cavity. The gallbladder is on its inferior surface. The pancreas is located posterior to and just slightly inferior to the stomach. Those two connect to the duodenum. The pancreatic duct and the bile duct coming from the liver gallbladder come together and open up into the duodenum, where they can deliver their digestive juices. Like I said earlier today, the liver makes bile, which is an emulsifying agent for the lipids that we eat. And then the pancreas makes digestive enzymes that are used in the small intestine. Most of the enzymes that are active in the small intestine come from the pancreas. Some are made in the small intestine, the brush border enzymes I mentioned earlier. But there are a lot of enzymes that come from the pancreas to work in the small intestine. This is, I don't want to get into the structure of this too much, but this is just a suggestion of the structure of the liver. It's a huge organ that has a repeating structure in it of these things called lobules. And it's a very homogeneous organ. One area looks exactly like the other. It's organized around these filtration structures that basically filter blood through to clean it out somewhat before it leaves the liver and goes on in circulation. As it's filtering that blood, which is going to remove toxins that we might have ingested or something like that, it will also remove components that are used as bile. And then the bile is collected in these light green ducts that we see here, which eventually collect and deliver the bile out to the small intestine. That bile collects and is delivered through the bile duct or common bile duct, which we see here, and it meets up with where the pancreatic duct opens up into the duodenum. There's actually a separate accessory pancreatic duct that bypasses the bile duct if there's ever a need for pancreatic secretions to get into the duodenum separate from bile. But largely the two release their contents at the same time at the same place down here. I already talked about the liver, in the pancreas, talking more about the islets, but the stuff that we didn't talk about, all of the cells make up the pancreas that aren't part of its endocrine function, make those enzymes and secrete them through the pancreatic duct. And then, like I said, I tend to include the gallbladder when I'm talking about the liver. In the book, they separate it out. So talked about the liver, mentioned the pancreas, now we're back to the gallbladder. I pointed out the common bile duct a second ago, which we see here. It's an extension off of where the hepatic bile duct meets up with what's called the cystic duct. And the cystic duct is connected to the gallbladder. When bile coming from the liver drains down through the hepatic bile duct into the common bile duct to that opening, if the opening is not in fact open, then the bile will build up in the bile duct and backfill through the cystic duct and be stored in the gallbladder. Now, it's called the cystic duct because the word cyst really is another word for bladder. We don't use it too often to mean that. When you hear cyst, you don't think of bladder. You usually think of something that's kind of like a benign growth or something like that. But that's where the word comes from. And I mention that because there's a hormone released from the duodenum called cholecystokinin. And let me just write that word down real quick. Cholecystokinin. And it has in the center there cysto, which refers to the bladder. Chole is the Greek word for bile or gall. Gall and bile are the same thing. We don't tend to use the word gall too often to refer to bile, but that's actually what the word means. So bile or gall, bladder, which is what cysto refers to, and then kinin comes from the Greek word that means movement. We refer to cholecystokinin just by its abbreviation, CCK. Any situation where you have to refer to that, please use the abbreviation. But I'm telling you the name because its meaning is important. This hormone is named gallbladder movement, essentially, because that's what it does. When it's released from the duodenum, it causes the gallbladder to contract, and that'll push the bile that's stored in the gallbladder out into the duodenum. That's going to happen as soon as food stuff moves from the stomach into the duodenum. So anytime when the chyme is hypertonic, especially the fact that it has lipids in it, that'll stimulate the release of CCK from the duodenum. It's a hormone, it'll move through the bloodstream, it'll get to the gallbladder, and it just stimulates the gallbladder to contract. And it'll squeeze all the bile out. all the bile that's stored up since the last meal is released all at once to help emulsify the emulsify the lipids in the food that you've just eaten. The liver will continue to make bile and it'll slowly leak out through the common bile duct as more food's moving on from the stomach. So that initial burst of bile will mix in with the food as it's coming out of the stomach. Now, the body likes to repurpose things quite a bit. So CCK does a little bit more than just stimulate gallbladder contraction. Let me just, activated by hypertonic chyme, it causes contraction of the gallbladder, which will release that bile, like I was just saying. It also stimulates the pancreas to make more enzymes. So if you have hypertonic chyme, that means there's a lot of stuff that needs to be digested. And CCK, while it tells the gallbladder to contract, also tells the pancreas to go ahead and start making enzymes, more enzymes, to help digest all that food that was just released from the stomach. There's also an enzyme called secretin. I'm not going to talk about what the name means too much. It's kind of obvious. It's not a very inventive name for an enzyme. But this is activated by acidic chyme. And it causes the liver to make more bile. And it causes the pancreas to secrete bicarbonate. So there are a number of enzymes associated with the small intestine, duodenum area, but these two I think are important to mention. We already talked about gastrin in relation to the stomach, but CCK and secretin are released. When chyme moves out of your stomach, depending on what it's made up of, it'll activate the release of these two hormones. The more hypertonic the chyme is, the more stuff there is, the more macromolecules dissolved in the chyme, the more CCK that will be released. The more acid in the chyme, the lower the pH, then the more secretin will be released. Now, chyme is rarely, if ever, going to be just hypertonic or just acidic. If the stomach is active, then it's going to be releasing acid, and whatever food's being broken down, those macromolecules will be present in the chyme. So really there's going to be a mixture of the two. It's just we can see a little separation in what the two end up doing. Okay. Now, bicarbonate we've met before. That's the HCO3 minus ion that we've seen before. But where we've seen it is in relation to helping to buffer the pH of the blood. So in the red blood cells, it's produced having to do with carbon dioxide transport. We just saw today that in the in the parietal cells, it's produced to make the stomach acid, and the bicarbonate then leaves that parietal cell and goes into the bloodstream to help buffer the blood. The pancreas makes bicarbonate also. This bicarbonate, however, is used to buffer the stomach acid. The pH in the stomach when it's active is going to be pH of 3 or 2 or maybe even lower. For the small intestine, that pH has to be neutralized very quickly back to 7.3 or 4, where it needs to be in the small intestine. So the pancreas releasing bicarbonate into the duodenum will neutralize the stomach acid very quickly as it gets into the small intestine. I pointed out earlier that there is an accessory pancreatic duct that bypasses where the common bile duct connects. And all I can imagine is that accessory pancreatic duct would be a route to get bicarbonate into the duodenum quickly by itself, especially when, if you say have a lot to drink, your stomach's going to produce acid, but there might not be a lot of macromolecules present. And so there'll be a more secretin being released and more bicarbonate stimulated from that. That's more important than the gallbladder or the bile from the liver, really. Now, bile does actually have some bicarbonate in it also, but the majority of the bicarbonate that neutralizes stomach acid in the duodenum comes from this pancreas. So that's really the highlights of what you need to get out of the liver, gallbladder, pancreas stuff there. And the last section of the book is about chemical digestion and enzymes, which I've already talked about. I prefer talking about that at the beginning. So I did that a little bit last week and a little bit at the beginning of today, which finishes out the digestive system chapter. After the digestive system chapter, however, there is the metabolism chapter. And I want to talk about this a little bit. Now, I want to start off by sort of explaining to you what you don't need to bother getting out of this chapter. This poster that I keep on my cart now, I used to have it up on my office, but I bought some artwork and replaced it. This is one of two posters that represents all of the chemical reactions that take place in cells. Now, probably no one cell in the world does all of these things. In fact, this is going to represent the chemistry that takes place in the cells in our bodies and chemistry that takes place in cells in plants and that sort of thing. So it goes beyond just individual types of organisms. It's sort of everything. But the word metabolism really is referring to all of these reactions. Okay. What I want to refer to for metabolism is less than that. It's just about how we make use of carbohydrates to make ATP. Now, in this chapter, they are kind of addressing that whole poster, not in that much detail, but they have an overview of metabolic reactions and then separate sections on carbohydrates, lipids, and proteins. And then some metabolic states of the body stuff, which is going to tie in a little bit to thyroid hormones and adrenal cortex steroids. Also, energy and heat balance are included there. I just want to go into the carbohydrate metabolism and talk about that. Now, metabolism is made up of two sort of opposing concepts. There's anabolism and catabolism. Anabolism means to build up, catabolism means to break down. And all we're interested in here is how we break down glucose to get energy. So really all I'm interested in you understanding about metabolism is carbohydrate catabolism. Okay. And there's it's represented in this picture, which is essentially the same as that chemical reaction that I showed you at the beginning of the respiratory stuff last week. The CH2, oh, sorry, I said it wrong. C6H12O6 plus 6O2 yields 6CO2 plus 6H2O. That equation that I showed you last time is essentially what this is getting at. Here's C6H12O6, a glucose molecule. It's broken apart into two pieces. Those pieces get modified, sent through a series of reactions. All of those things harvest energy, and that energy drives the production of ATP. Okay. This is essentially what cellular respiration is. Now, truthfully, the reaction that I showed you, the C6H12O6 reaction, is only addressing the top three parts of this picture. The last one is separate from that reaction that I showed you, but that's where the energy comes from. We can't have that one reaction take place all at once. The amount of energy that would be released by completely breaking down glucose in one step is essentially a fire. That's what's happening when wood burns. Wood is made up of cellulose, which is just a bunch of glucose. In the presence of oxygen, it's burning and it gives off all that heat and light. We don't want that happening in our body, so really that reaction is broken down into a series of smaller reactions. The first set of those fall under what we call glycolysis, which literally means breaking glucose. So the 6-carbon glucose molecule is broken into 2 3-carbon molecules called pyruvate. Pyruvate is then modified slightly. One of the carbons is given off as carbon dioxide, and the other 2 are attached to a carrier molecule represented by this circle. And that is introduced into a cycle of reactions here called the Krebs cycle, or the citric acid cycle. It's another name for it. In the citric acid cycle, the rest of the carbon from glucose is given off as carbon dioxide, and then energy is being harvested. The energy that's being harvested, while some of it does directly go to making ATP, represented in glycolysis, the Krebs cycle here, just a little bit of ATP, most of that energy is harvested in the form of electrons that we have control over. And they're sent through a series of proteins called the electron transport chain that are used to power the production of ATP. That's essentially what cellular respiration is, and that's what these pictures point out. I want to show you a couple of pictures that give a little bit more detail. This is a bit more detail to glycolysis. It's really made up of like 9 different chemical reactions, which are not all represented here, but this sort of gives a bit more detail about what's happening. Glucose is split, a process that requires phosphates added into it. We phosphorylate the glucose, which is represented by these little yellow balls here. They're broken apart. It's breaking it apart harvests a little bit of energy out in the form of ATP. Breaking it apart causes the creation of a couple more phosphates. And then energy is harvested out as electrons as things are broken down. Those phosphates that we see are used to reconstitute ATP, in the end making 4 ATP molecules. But we needed 2 ATP molecules to get started in the first place. So the net gain from 1 molecule of glucose is 2 ATP molecules. But there's a good bit of energy in the form of electrons that are harvested also. Okay, and we have the 2 pyruvate molecules. Pyruvate then moves on into the mitochondria, where the Krebs cycle takes place. Now, I like the level of detail that previous picture shows you, which is mostly represented by hexagons and half hexagons to represent the shape of carbon molecules. This picture goes into more detail than I care for you to know. Don't worry about all these chemical structures that are indicated here. The point of this is just that carbons harvested from glucose are introduced into this series of reactions, which are all carbon molecules being modified here and there. Out of that comes some CO2, so there's one, there's another, and a phosphate that is used to make essentially an ATP. And then I think it's where we see these yellow circles here, I'm not positive. electrons are harvested. And then eventually the electrons are used, pass through these series of proteins here called the electron transport chain. These proteins are found in the inner mitochondrial membrane. Okay? So there's an outer mitochondrial membrane enclosing the entire mitochondria. The Krebs cycle reactions are taking place inside the inner mitochondrial membrane. And so those electrons pass through this series of proteins here, which using the energy from those electrons, pump hydrogen ions across the inner mitochondrial membrane so that the concentration of hydrogen ions is quite high between the inner and outer mitochondrial membranes. The only way for the hydrogen ions to get out is to pass through this big thing here, which is called ATP synthase. The physical movement of hydrogen ions through this enzyme powers the chemical reaction that sticks a phosphate onto ADP, making ATP. There's not a perfect one-to-one correspondence of electrons and hydrogen ions to ATP production, but there is a particular relationship there. There's a certain number of ATP that'll be made through this process. At the end, the electrons end up getting combined with hydrogen ions that are coming out here and oxygen to make H2O. Okay, so in the process here, we've seen C6H12O6, glucose, getting the whole thing started. Oxygen's added in here, and we get out of it CO2, which we saw in a few places, and water, which we see here. So that accounts for all of the players in the reaction that I showed you before. It's just a very involved process getting to all of those. And in doing that, we make a whole bunch of ATP. This is essentially the same picture at the beginning, but here the numbers are a little bit more specific. Glycolysis generates, for each glucose molecule, 2 ATP molecules. The Krebs cycle, from 2 pyruvates, which equal a single glucose molecule, generates 2 more ATP. And then all of the electrons that are harvested are kind of tallied here. They go through the electron transport chain, and they generate another 34 ATP molecules based on that original glucose. So 2 plus 2 plus 34 gives us 38 total ATP. There is a little bit of variation in the system. Sometimes it might only make 30 or 32 or so ATP overall, but it can make up to 38 ATP molecules per glucose molecule. That's essentially what that's getting at. That's the basis of what I want you to get out of this metabolism stuff. Glucose going through all these steps gives us a whole bunch of ATP. Requires oxygen for the mitochondria to run the Krebs cycle, and we're harvesting most of that energy is electrons, which power the electron transport chain and ATP synthesis. The next part in the chapter here goes on to other carbohydrate metabolism things. You don't need to look at that stuff. I don't care about that. I just want you to get a feel for what's going on in glucose being used to make ATP. And then the rest of what's in this chapter about lipid metabolism, protein metabolism, metabolic states, energy, et cetera, et cetera. You don't need to get into. I do want to say a couple of things about nutrition, some of which is in the book and some of which I need to add to it in a second. There is a balanced nutrition that's required to get the proper nutrients into your system. The current representation of that is this My Plate icon. It used to be the food pyramid, which you might be familiar with, that sort of thing. But that gets at the macronutrients, the macromolecules that we need in our body, lipids, proteins, carbohydrates, that come from these various food sources. We also need vitamins and minerals, which we would call micronutrients, which are discussed a little bit here. What I wish was discussed in here, which are actually in the macronutrient section, are essential nutrients. So let me talk about that a little bit, which is basically to say nutrients that we have to get from our diet. Metabolism gives us the ability to create a lot of molecules that we use in our body. But there are some molecules that we cannot make in our body, and we have to get from somewhere else, and those are the essential nutrients. The essential amino acids are amino acids that we need to get from another source. They're usually complex structures like the ring structures of tyrosine or tryptophan, those sorts of things, or they contain elements like sulfur, that kind of thing, that need to be incorporated by some other organism. We can't do it ourselves. Depending on exactly how you define it and what source you're looking at, the number of essential amino acids varies. I think it can be as few as 9 or as many as 13. Our need for amino acids changes as we age. The number of essential amino acids that children needs is different. I think that's where the 13 essential amino acid comes from. And as we get older, we develop the ability to make some of those amino acids on our own, and only a subset are needed in our diet. If you don't get these essential amino acids into your diet, you will not be able to make all of the proteins that you need in your body. That's a concept that's called protein load, just what your body needs to make for proteins. And you need to have the amino acids available to do that. If you transcribe a gene into mRNA to make a protein, it goes out into the cytoplasm, and the ribosome is reading that mRNA to make a protein, and it gets to a point where it needs an amino acid, that amino acid isn't there, the ribosome will just stop making the protein, and you won't be able to make protein anymore. And so having the essential amino acids is vital to a lot of the processes in your body to take place. The easiest way to be sure that you get the proper essential amino acids in your diet is to just get them from another organism with a similar protein load. Protein load is a fairly broad characteristic. Animals all have the same protein load. So really eating animals is the best way to get all the amino acids that you need to in your body. You don't really have to think too hard about it. If you eat proteinaceous animal sources, meaning meat primarily, you'll get all the essential amino acids in your diet that you need. Of course, there are a lot of people that would rather not do that for various reasons. If somebody is a vegetarian or a vegan, they need to pay special attention to where they get essential amino acids from. Neither by themselves is a great source, but when you put grains and legumes together, they make for a great overall essential amino acid source. Legumes are beans for the most part, and so simply rice and beans is a great vegetarian meal. And if you are a vegetarian or vegan, hopefully you already know that. If you don't, then you have bigger problems. But something like beans and rice, great way to get essential amino acids from a vegetarian source. Variations on that will work just fine. I suppose corn and peanuts would do the trick too. Okay. Corn being grain, peanuts being legume would do it. It's a very odd combination. I don't know that people tend to eat. Well, I guess that'd be Cracker Jacks, wouldn't it? Cracker Jacks is a great source of essential amino acids. Yeah, so any combination of those sorts of sources together make for a very good source of the essential amino acids that we need. There are also essential fatty acids. And they get a good bit of press these days, or I should say marketing these days, as food companies are trying to entice people to buy their products. They'll highlight the presence of essential fatty acids. The essential fatty acids are collectively called omega-3 or omega-6 fatty acids. Technically omega-9 also. All those are fairly rare. Let me just pull up a picture of a fatty acid and talk about that for a second. Unfortunately, when you do a search for this, you'll get a lot of stuff about nutritional supplements. So obviously, this is more about advertising stuff. But here's a picture that shows this. Now, fatty acids are hydrocarbon chains, carbons and hydrogens, and a chain-like structure with this kind of structure at the other end. So there's a carbon double bonded to an oxygen and bound to a hydrogen-oxygen pair here. This COOH ending is what makes it an acid. Okay. The rest of it is just the fatty tail. Okay. The end that has the COOH on it is the alpha end of the fatty acid. And so the other end is the omega end. Now in this figure here, you can see that the omega end is actually labeled methyl end, which is a chemical aspect. But calling it an omega-3 or omega-6 fatty acid is because this end is the omega end. It's the opposite end from the alpha end. There's a carbon right here, then there's a carbon here, and a carbon, 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 carbon. The lines between these carbons represent bonds. When there are two lines between when there are two lines between a pair of carbons, that means that there's a double bond there. Starting at the omega end, the first bond is single, the second bond is single, the third bond is double. That's an omega-3 fatty acid. If the first bond is single, second bond is single, third bond is single, fourth bond is single, fifth bond is single, sixth bond is double, that's an omega-6 fatty acid. Now, this omega-3 fatty acid actually does have a double bond in its sixth position, but that's not an omega-6 fatty acid because the third bond is already a double bond. Okay. So that's the difference between omega-3 and omega-6. We don't have the ability, we don't have the enzymes in our body to put double bonds in those positions in fatty acids. But we need fatty acids with double bonds in those positions, especially to make membrane phospholipids. So getting omega-3s and omega-6s into your diet is important so that you have the proper materials to make membrane phospholipids. And if we just look a little bit further at the pictures in the image search that I just did, you can see some good examples of sources of omega-3 fatty acids. So one is fish. A lot of the pictures that have pills in them, these are fish oil pills. But you know, salmon steak would be a good choice too. This bowl, I imagine, is supposed to represent flax seeds. Flax is a great source of that. Flax seed or flax oil. Looks like those are walnuts, maybe. Eggs. I imagine that's supposed to be olive oil. This is seaweed. I guess that's kale there. Those are all sources of these types of essential fatty acids. So you really do need these things in your diet to make proper membrane phospholipids. Now, a very important part of getting those into your diet is if you're pregnant. for the proper development of neurons, and because membranes and neurons are so important for regulating action potentials, that sort of thing, having these essential fatty acids in your diet if you're pregnant is crucial. So most prenatal vitamins will have something like DHA included. DHA is one of these types of fatty acids. And that'll help with development of neurons because of how it helps with the cell membrane stuff. Now, technically, there is an omega-9 fatty acid, but they're not quite as crucial, but you might see reference to them now and again. 3s and 6s are the main ones. So I just want to add that in. I kind of wish that the book would just have a little section. Okay. So up here with, whoops, where'd it go? With the MyPlate thing, it's sort of getting at sources of macronutrients that you need in your diet. They could simply just add after that some things about essential nutrients, but then they get into vitamins and minerals there, which I'm scrolling through quickly kind of to represent. I'm not going to get too hung up on that stuff. It's the essential nutrients that I want to stress there. So that's what I wanted to include about metabolism and nutrition, just to pull that out of this chapter here. But I think it fits in with the digestive system stuff quite well. Oh, sorry, I want to pull something up real quick. I skipped over this section that was chemical digestion, closer look, because I talked about enzymes quite a bit already, but I want to point out a couple of things. This is just sort of an overview of all of the different functions in the system and where they're kind of taking place. This is the table of enzymes, which I was talking about. Remember, I was talking about lingual lipase and salivary amylase and those sorts of things. By the way, sucrase, maltase, lactase are all in here. Let's see. This is an overview of where we absorb water. Water is introduced to our system, of course, as we eat or drink, because there's water in our food and obviously in what we drink. Plus, we secrete lots of water in the system: saliva, gastric secretions, bile, pancreatic juices, intestinal secretions, both small and large. In the end, only about, 150 milliliters of water comes out as part of our feces. So a lot of the water that we ingest and that we secrete gets reabsorbed. Most of that happens in the small intestine, but some of it happens in the large intestine also. If you have intestinal discomfort, some sort of infection that's wreaking havoc with your intestines, a common symptom to go with that will be diarrhea, because as the intestines are fighting off an infection, they're not going to be absorbing water very well, so the amount of water that comes out at the end is going to be much higher than normal. And so often when people have diarrhea, they need to keep themselves hydrated because they're not retaining as much water as they should. One last thing I want to mention, actually going back to the small intestine stuff, is about absorption. This is the only picture that really addresses absorption, but I want to point out this and then a couple of other things. This is just about lipid absorption. Lipids in the form of fatty foods get broken down into fatty acids and other small lipids that just diffuse across the cell membrane unimpeded. They're lipids, cell membrane's lipid-based, so they just move right into the cell. They get packaged up, for the most part, into little structures called chylomicrons, which the book, well, it says right there. It should say somewhere up here. I don't see it. Oh, there it is. Chylomicron. And that's just a little protein structure that's holding on to the fatty acids. And those move into the lymphatic system. So this is a lymph capillary here. Remember that one of the functions of the lymphatic system is to transport dietary lipids. And that's where it's happening. They get absorbed out of the small intestine. They get packaged up as chylomicrons, and then they move into the lymphatic system. They don't represent in a picture form here the absorption of amino acids or carbohydrates, but I do want to say a little bit about that. We tie the movement of those hydrophilic molecules across the membrane with various active transport processes. We don't actually actively transport the nutrients across the membrane, but we expend energy usually on building up sodium concentration gradients and use the movement of sodium across the apical membrane of the intestinal wall to help pull amino acids and carbohydrates in also. There are sodium co-transporters for amino acids and monosaccharides, which move them into these cells, and then they diffuse across the basal membrane into the bloodstream. It's not represented here, but that's the sort of thing that happens. So I just wanted to go back and pick that up. I forgot to mention it before I got into the metabolism stuff. So that's the digestive system. I want to move on to use the rest of the time we have today to start talking about the urinary system, like I started with the digestive system last time. Next week, which will be our last full class period, on the syllabus it says urinary and reproductive systems, and it's just not possible to do both of those all in one day. So I want to start it today. What I want to do today is just highlight things I want you to pay attention to in the urinary system stuff. What we're going to do next week for the first half is to look at urinalysis, and in that understand a little bit more about what the urinary system is doing. And then in the second half next week, we'll look at the reproductive system, which we'll pay most attention to how reproductive cells are made, where they're made, and how they're brought together between the male and female reproductive tracts. So that'll be the focus of what we're doing next week. So what I want to talk about today, I'm going to skip over the physical characteristics of urine because that'll be something that we talk about in regards to urinalysis next week. I'm not going to talk about this too much, but I do want to point it out. Oh, sorry, I forgot the way the book lays this out. I do want to talk about this. This is the thing I said earlier that I was hoping to get to before the end of the day, which is about urine transport. And really this should say urine transport and elimination, which is why it kind of caught me by surprise. And I don't quite understand why the book lays this out. It, to me, seems backwards. So I'm actually going to go down to the bottom for a second and deal with stuff. The ureters are the tubes that carry urine from the kidneys out. They carry it to the bladder, which we see here, where it's stored until we have a sufficient volume to get rid of. And we void our bladder reflexively, very much like I was describing for the defecation reflex. But I want to describe this a little bit more. Now, in a book like this, they're going to talk about the micturition reflex, which is the voiding reflex. But it's a little bit more involved than that. So, there's two aspects of what happens reflexively. One is what I like to call the holding it reflex. As the bladder starts to stretch, it signals the sympathetic system to contract the internal urethral sphincter. Remember, sphincters are contracted by the sympathetic system. Okay. I usually talk about that in terms of the digestive system, but it's true of the urinary system also. Okay. The sympathetic system also keeps the detrusor muscle. Better check the spelling of that. Hold on a second. Yep, I spelled that right. The detrusor muscle, which is the wall of the bladder, relaxed. Now, you cannot actively relax a muscle. Okay? Muscles, when they're activated, by definition, contract. What you can do is you can inhibit the activation of a muscle. And that's what the sympathetic system does. Okay. Now, this is when the bladder starts to fill, when the wall starts to stretch. When it's empty, the stretch receptors are essentially silent. And then as it starts to fill, there will be low-level activation of the stretch receptors because this is starting to take place. And so the sympathetic system keeps that sphincter constricted so that you don't void your bladder. Okay? And the detrusor muscle stays relaxed because of sympathetic influence. Again, to keep, so that you hold the, or store the urine in the bladder. Then the rest of this is the voiding reflex, which is what the book calls the micturition reflex. And I'm not saying that it's not the micturition reflex. I'm just giving it a special name because I want to talk about the difference between holding versus voiding. And so this is when the bladder is full, then the stretch receptors are very active. It signals the parasympathetic system to contract the detrusor muscle. It also then relaxes the internal urethral sphincter. Now, I put relaxes in quote again because you don't really actively relax a muscle, but it does keep it from being contracted. You have voluntary control over the external, can't type, external urethral sphincter, which is skeletal muscle, okay, just like the external anal sphincter. While I suggested it might be true in the digestive system, is definitely true in the urinary system, that the parasympathetic division can override voluntary contraction of the external urethral sphincter. When you gotta go, you gotta go. What that means is when the bladder's full, and there's nowhere else to store urine, the parasympathetic system will say, sorry, can't hold anymore, and your voluntary control of the external urethral sphincter will be lost. If that weren't to happen, if the urine filled the bladder, it's going to back up into the ureters. The ureters also can hold a little bit of urine. They have structural similarities to the bladder. But once they're full, if the urine then is going to back up into the kidney, you're going to create all sorts of problems. The function of the kidneys is based on maintaining very important extracellular fluid concentrations. And if the urine backs up into that, it'll throw those concentrations off, and the kidneys will stop working. So you get kidney failure, which is pretty much a death sentence. If you can't filter waste products out of the blood, then you're going to have all sorts of problems ensue. If you've ever seen House, a few episodes of House, they're not talking about neoplastic syndrome. They're going to talk about kidney failure or liver failure. Waste products build up in your system, your body's going to die. And so the parasympathetic system can override your personal decision to not go to the bathroom, and the external urethral sphincter will relax and you'll go. It seems like a similar kind of control should be present in the digestive system, but I've not found a clear source saying that's the case. But it's not the same problem. You can't really have the digestive system back up and cause the same kind of life-threatening problems as what would happen in the kidney. But an impacted colon can rupture, and then you'd be septic and you'd die from that just the same. So there might be something similar in the digestive system, but definitely in the urinary system. One very unusual situation in the body where the parasympathetic system, the autonomic system, has ultimate control over so-called voluntary muscle, skeletal muscle. It doesn't actually do something to the muscle itself. It instead inhibits the motor neurons that control the muscle. And so you don't have the ability to contract that muscle, and you'll void your bladder. So that's a little bit more in detailed description of what's happening, but the book talks about the McPherson reflex here, and they're kind of getting at it. The book will leave out a lot of the holding it, the sympathetic nervous system put to the system, but it is in fact there. Okay, so I wanted to point that stuff out. It kind of fits in with the digestive defecation reflex I talked about earlier. I'm not going to go into the gross anatomy or the microscopic anatomy of the kidney in a whole lot of detail. Next week, when we're looking at models that'll go with the urinalysis lab material that we're doing, I'll talk about that in a little bit more detail. But you ought to look through this stuff a little bit before you come in, so that you kind of know where this is going. The sort of functional part of what filters blood in the kidneys is this structure here called the nephron. The nephron is actually the kind of yellowish stuff in this picture. It's surrounded by capillaries, and the nephron and the blood supply going through it, the capillaries surrounding it, work together to help regulate what's in your blood and what's going to be eliminated as urine. And that's what's happening there. Here's a bigger picture of the nephron. The nephron actually goes up to about here, and then the rest of what we have, this tube here, which has several sort of truncated branches attached to it, is where other nephrons come together, and then the urine collects in this duct, which is called, inventively enough, the collecting duct. And then it leaves the kidney and is stored in the bladder. The next section is just talking about the cellular structure of these things a little bit more in detail. the capillaries and other structures that help to filter the blood, what regulates what's going on in the kidneys, a little bit there. Then it gets into the physiology of the urine formation, what's happening in the tubes of the nephron, and then how we regulate blood flow, which is mostly through blood pressure, but also a little bit through blood volume. And that's handled mostly by the kidneys. And then endocrine regulation of that stuff. The kidney releases an endocrine a hormone that plays a big part in regulating blood pressure as part of kidney function. And then some things about regulating fluid balance and that sort of stuff. After talking about the digestive system, I took a second to go into the metabolism chapter to talk about that stuff in a little bit more detail. Following the urinary system, there's another chapter that's called fluid, electrolyte, and acid-base balance. I'm not going to go into that chapter the same way I dipped into the metabolism chapter, but so you can kind of skip over that. Any of the stuff that I want to cover is in this chapter. Those last few sections there referred to. The main point I want to get out of this actually is how the body has solved similar problems between the digestive and urinary systems. And I think it's in this section here. This figure shows a highly schematized cell that's in the wall of one of the kidney tubules. This side of the picture is the lumen of that tubule. So this is where the stuff that's been filtered out of the blood is located. What's going to eventually be urine. We don't call it urine until it gets to the collecting duct. And then obviously this red thing here would be a blood vessel. And this picture is just showing all the different ways that we move things across this tissue wall. We have a sodium-potassium pump, which uses ATP to pump sodium out of the cell. Potassium happens to come in. This is taking place to build up a sodium concentration gradient. By removing sodium from the cell, any sodium that's in the filtrate, in the lumen of the tubule, is going to want to move into this cell. It'll eventually be pumped out, move into the blood, which will be filtered in kidneys again. So the sodium concentration out here and the sodium concentration in the blood are roughly the same, and the sodium-potassium pump is basically making that concentration lower inside the cell. Sodium is used to move other things across the membrane. There is a co-transporter that pulls chloride in with the sodium or calcium in. Notice both of those when sodium is pulling one of those other ions in, hydrogen ions are being pumped out. Okay. There's also a sodium hydrogen antiporter represented here. So we can pull some things in, and all three of these are getting rid of hydrogen ions, which is going to get rid of acidic pH in various places. Here we see sodium tied to pulling amino acids in, or sodium tied to pulling glucose in. Hopefully that sounds very familiar. When did I say that before? How quickly you forget. Probably a half hour ago. I was talking about this in the digestive system. Okay. book had a picture of lipid absorption. And when I showed you that picture, I then went on to say something about amino acid and carbohydrate absorption. There's not a picture of that in the digestive system. But it is essentially this mechanism right here. When sodium is pulled inside because of the sodium concentration gradient built up artificially, it pulls glucose or amino acids in. It's happening in the digestive system to get the nutrients from the food you've ingested into your body. It's happening in the urinary system to get those things out of the filtrate. The filtrate filters everything out of the blood, okay? Even the things that you don't want to remove from the blood. So those things get pulled back in. So amino acids and glucose, they get filtered out of the blood by the kidney, just get reclaimed back into the blood. Okay. Whenever there's something wrong, like if you have too much glucose in your blood, hyperglycemia, this mechanism won't compensate for that. So you'll have some glucose in your urine, which is a symptom of hyperglycemia or diabetes. It's essentially the diagnostic criteria for it. But under normal conditions, any glucose that gets filtered out in the kidney just gets reclaimed here using the exact same process as the digestive system. Okay. And really, if you think about what's going on in the digestive system, we need to get other micronutrients like chloride or calcium or phosphates or magnesium or whatever. Those things are moving out of the filtrate into the bloodstream here. Again, the exact same mechanism as in the digestive system. You already have the genes for the proteins that do this. There's no need to reinvent the wheel. Just reuse them here. Probably in some ancient organism, there was not really a difference between the two systems. Maybe, I'm not that clear on, I can't really defend that claim too much, but if you think about a sponge, which is kind of the most simple animal there is, they don't have a digestive system and a urinary system. A sponge is just essentially a tube that seawater moves through, and it moves stuff out of the seawater and into its cells. It doesn't even have a bloodstream for that matter. But it probably has the same kind of proteins in its system to get that stuff out of the seawater and into its body. We've just diversified cells to maintain a couple of different processes, whether it's digestion or the waste disposal of the urinary system. But both cells use the exact same genetic tools to get the exact same job done. We just see that repeating over and over. While renal failure is a very important issue and is the cause of most of the drama in your average ER or house type show, I don't really care to get too much into the urinary system, nuts and bolts. I think this is the real take-home message, that the urinary system is, well, sorry, I should say that a little bit. There's, what we're going to do next week with the urinalysis is an important understanding of what's going on. I'm going to go into that detail here, but the thing that I'm not going to be able to talk about in terms of the urinalysis stuff is this idea that the system is using the same mechanisms as other systems in your body. We probably have a tendency to think of what the urinary system does as kind of, gross. We'd rather think of it as separate from everything else in our body. Okay? But really, it's just using some of the same mechanisms that are present. And I think this picture kind of goes some way to kind of illustrate that. I really wish that the book had a similar picture showing the same kind of things for the digestive system. I would imagine that the chapters on digestion and urinary systems in this book were not written by the same person, so that connection wasn't quite made. But I think that's an important thing to consider here. Another thing to be aware of in this section here, this picture shows something that we should be familiar with by now. H2CO3, carbonic acid. Carbonic anhydrase can make carbonic acid out of CO2 and H2O. It dissociates into hydrogen and bicarbonate. In the previous picture, I showed you a bunch of different ways that hydrogen ions move out of the cell. And if we do that, if the hydrogen ions get into the urine, that means that they're getting out of our system, or we're helping maintain the pH of our body. Okay. The bicarbonate that comes from making this also is available to move into the bloodstream to help regulate blood pH. Okay. So in the red blood cells, we saw the carbonic anhydrase reaction, and bicarbonate there helped to buffer the pH in the plasma. Earlier today, I showed you the parietal cell in the stomach making stomach acid, and the bicarbonate that comes from making the stomach acid goes into the bloodstream, helping to buffer the blood. And then here in the urinary system is trying to help acid-base balance. And so it can do that through the carbonic anhydrase, partly by getting hydrogen ions out of the system and into the urine, and getting bicarbonate ions into the plasma to help buffer. Now, the nice thing about buffers is they work when you need them. And if you don't need them, they don't do anything. If you have acidic pH in your blood, the buffer will get it back to neutral. But if you have a neutral pH, the buffer is not going to make it basing. It's not going to just move the pH to a higher number. That's not how they work. They just get things to the neutral pH for the most part. So this kind of thing's happening all the time. It's available to work at getting the pH in your system down if necessary, but any bicarbonate that's made that gets into the plasma is there to help buffer when it's needed, but if it's not needed, it doesn't have any huge effect on things. Now, there is another reason for making sure that hydrogen ions get into the urine, and that's that the urine needs to be a little bit acidic. The acidity of urine is going to inhibit microbial growth in the urinary tract. And so a little bit to help protect the urinary tract against infection, the kidney will get the pH in the urine a little bit low to help that out. So in doing that, it might produce bicarbonate, which will get into the bloodstream too. So, I'm not going to explain this too much, but this is one of the reasons why I really don't like getting into a whole lot of detail about the urinary system, this counter current multiplier system. This is just explaining how the kidneys concentrate waste products in the urine so that we can clear them out of our body efficiently. It's dependent on maintaining some high concentrations in the tissues of the kidney. If the urine backs up into the kidney, like I described, you don't want to have happen, it throws this off. And this keeps the kidneys from being able to do their main job, and you're not able to clear waste out of your blood very efficiently. Take a look at this. I usually spend some time trying to explain it, and truthfully, I don't know how much that explanation helps or confuses students. So this time, what I'm going to do is not talk about it. Look at it, hopefully over the next week. If you have questions about it, please bring them up next week, and I'll try to clarify. It's not as crucial to understanding things about the urinary system as far as I'm concerned as the other stuff I've highlighted here. But I do want you to look at it. And I'm not going to talk about this system but I'm going to talk about why it's important next week in terms of the urinalysis stuff. Yeah. So those are some, I wanted to hit some important topics there. Now, next week, we're going to be doing urinalysis. If you are in the habit, before you come into class, every morning to stop by the restroom, please don't. Come by the restroom. Come by the classroom first, and I want to give you a sample cup. Really? Okay. So you'll be performing your analysis on your own urine, which I'm sure is preferable to doing it on somebody else's urine. So please come in to the classroom first next week. It will be a whole week before I see you again, so hopefully telling you about it now, you'll remember it for next week.
And remember to drink a lot of water.
Yeah, and yeah, get a water bottle or something and bring that in the morning. I'm usually here about 7:30 in the morning, so, and I come straight here. I don't spend too much time in my office. So I'll be in here. I'll have the sample cups ready if you want to come in and get it right away. If there are people that are coming in right at 8 o'clock or need to, think about waterfalls for a while or something like that, there will be time factored into how we start out at 8 o'clock for people to get their samples. So, but I'm seeing people getting in a little bit later and later each week, since next week is kind of the last week. It's the last chance you have to come into class late. Please don't. You need to get your samples. So if you can get in before 8 and collect your samples before 8, that just means things will move faster. But if you can't get in until 8, there will be time for you to collect your sample. That's all I want to cover today. I could spend a little bit more time talking about the urinary system, but I'd rather save a lot of it to work into the urinalysis stuff next week. So we'll pick up with that, do the urinalysis in the first half, and then do the reproductive system in the second half. So, otherwise, have a nice week.